Hello, learners. How are you today? I hope you're all doing well. Last time, we learned about the respiratory system and circulatory system, together with a healthy lifestyle. Today, we will learn another lesson in Science 9, Module 2. But before we proceed to our discussion, let's check our most essential learning competency. And here it is. Explain the different patterns of non-Mendelian inheritance. Don't worry because I'm Jeanette Manyala, your science teacher, and I will discuss with you how all this happened. For sure, this will be another amazing lesson in Science 9. So, let's begin. In your grade 8, you learn about the basic of genetics, right? You have learned that the cells divide to produce two cells. Meiosis is one of the processes that produce genetic variation in Mendelian pattern of inheritance. You also learn about the Mendelian genetics and its three laws of inheritance, which are called the law of dominance, the law of segregation, and the law of independent assortment. But there are also patterns of inheritance that do not follow Mendelian laws or principles, and these were called the non-Mendelian pattern of inheritance. But before we continue the non-Mendelian patterns of inheritance, let us have a short review of some terminologies which can help you better understand the lesson. First, what is genetics? So, genetics defined as branch of biology that deals with the study of genes, genetic variation, and heredity among organisms. We also have the term inheritance. So, inheritance is a process in which genetic information is passed on from a parent to their offspring. What about variation? Variation is any difference between cells, individual organisms, groups of organisms of any species. Now, let's talk about gene. So, gene is a unit of heredity. It is a section of deoxyribonucleic acid that codes for a specific trait in an organism. We also have the genotype and the phenotype. So the genotype, it is, it is the actual genetic makeup of an organism, and it is a combination of two alleles. And that alleles can be homozygous or a heterozygous. But since we are dealing with the phenotype, it is the physical feature or appearance or trait of an organism. It is also the form of a given trait that is expressed or observable. For example, I have here a 4 o'clock flower. What do you think is the genotype of both flowers? So, the first flower is FR, and the second flower is FW. What about the phenotype of the first flower? Yes, you are right. It is a red flower. The second is a white flower. That is the difference between the genotype and phenotype. So, let us now consider the homozygous and heterozygous. Homozygous means having two identical alleles. Heterozygous means having two different alleles of a particular gene. So if we are going to consider the letters for the homozygous, it is a same letter. It can be a big letters or small letters that represents the allele. And then for the heterozygous, it is a combination of a big letter and small letter. So it is a combination of two different alleles. We also have a Punnett square. So the Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or breeding event. And did you know that Reginald Punnett is the one who created the Punnett square? So let's proceed now to the Mendelian patterns of inheritance. So it is a type of inheritance where in the patterns of phenotypes do not coincide with those that was presented in the Mendelian law of inheritance. First, we have the incomplete dominance. It is a form of intermediate inheritance in which one allele does not completely dominate another allele, which results in a new phenotype. An example of this can be observed in a snapdragon flower. A red snapdragon flower is crossed with a white snapdragon flower, results in a pink snapdragon. We can use the homozygous red flower as the genotype for the red snapdragon and homozygous white flower as the genotype for the white snapdragon. Crossing the red snapdragon and white snapdragon will result in 4 pink flowers. Each box of offspring in the Punnett square is equal to 25%. We can use this in computing the total probability of the offspring to appear in the breeding event. Let us identify the genotypes of the offspring in our example. Crossing red snapdragon with white snapdragon flower will result in 100% pink flowers. How about the phenotypes? The physical appearance of the offspring when we cross the red snapdragon flower with the white snapdragon flower is also 100% pink flower. Let us now proceed to the next pattern of inheritance, which is codominance. Codominance, both alleles are expressed equally in the phenotype of the heterozygote. For example, a red cow mate with a white cow will result in a 100% roan cow. Thus, both alleles or both appearances are expressed equally in the offspring. As you can see, this is a white cow with red patches. Now, let us have a short activity. You're going to identify the pictures presented and you are going to consider if it is incomplete dominance or codominance. So what do you think is the answer between chestnut brown horse crossed with a creamy white horse? As you can see, the resulting offspring is a palomino. So is it incomplete dominance or codominance? Excellent! You are right. It is an example of incomplete dominance. What about the cross between a standard poodle and a Labrador, resulting in an offspring called Labradoodle? Great! You are right with your answer. That is an incomplete dominance. For the cross between black chicken and a white chicken, the resulting offspring is a speckled chicken. What other patterns of non-Mendelian genetics are represented by the sample? You got it. That is codominance. Now, let's solve a given problem. Gray horse or GG are codominant to white horses or big W, WW. The heterozygous horse or GW is an Appaloosa, a white horse with a gray spots. Cross a white horse with an Appaloosa horse and give the phenotype and phenotype ratio n%. I'll give you time to solve for this. Now, the resulting cross produce a phenotype 2 Appaloosa horse and 2 white horse. So the genotype is 2 GW and the other one is 2 WW that represent a white horse and then the other one is an Appaloosa horse. Now, for the percentage, it is 50% Appaloosa horse and 50% white horse. Did you come up with the same result? Okay, very good. Let's proceed to the third non-Mendelian pattern of inheritance, which is a multiple alleles. So multiple alleles, an allele of a genetic locus having more than two allelic forms within a population. There are traits in human and other organisms that have three or more different types of alleles or genes. When a trait has three or more distinct alleles, we refer to it as having multiple alleles. The human ABO blood type alleles or trait is an example of a trait with multiple alleles. Three distinct alleles exist: allele A, allele B, and allele O. However, an individually only has a single pair of these alleles. The four main types of blood types are A, B, AB, and O. If you're a blood type A, your red blood cells have proteins attached to them known as A antigen. Type B blood carry B antigens. Here, type AB blood has both A and B antigens, and type O blood has neither A nor B antigens. Your immune system will produce antibodies against any blood antigens you don't have in your blood. That means people with a type A blood create antibodies against B antigens. A person with a type A blood receiving a transfusion of type B or AB blood would have an ABO incompatibility. So with that, with the reaction of incompatibility reaction, your immune system attacks the new blood cells and destroy them. If you have type AB blood, you have both A and B antigens. This means you are a universal recipient and you can receive any type of blood. However, you can only donate blood to other people who have type AB blood. If you have type O blood, which has no antigens, you are a universal donor. You can give blood to anyone without triggering any immune system, but you can only receive type O blood. Before a blood transfusion, your doctor will test your blood to determine your blood type. A small sample will be cross-matched with some of your donated blood. The two samples of blood are then mixed and watched for a reaction. This allows your doctor to be certain an incompatibility reaction won't take place. So our blood is very important. Blood typing is very important. Take note of that. Now, we have here a quote in rabbits. It is inherited as a series of multiple alleles. This means that there can be more than just two alleles for a single gene in the case of coat color in rabbits. There are four alleles, and each one is expressed with a different phenotype. So try to look at the figure here. So we have here a wild type, which is brown fur, and then we have here the chinchilla, which is a black tip white fur, and the other one is the Himalayan, which is white fur with black paws, nose, ears, and tails. And the other one is the albino, which is a white fur. So now let's solve a genetic problem. Suppose a newborn baby was accidentally mixed up in the hospital. In an effort to determine the parents of the baby, the blood types of the baby and two sets of parents were determined. Baby 1 had type O blood. Mrs. Brown has type B blood, and Mr. Brown has type AB blood. Mrs. Smith had type B blood, and Mr. Smith had also type B blood. Now, draw the Punnett squares for each couple. You may need to do more than one square couple. And then, to which parents does baby 1 belong, and why? Hint, you may want to refer to your Punnett square. Okay, so try to solve it in your paper so that we can determine who are the parents of baby 1. Now, by mere looking at our crosses here, so we have 4 crosses here. Okay, 2 crosses for Mr. and Mrs. Brown, and then 2 crosses for Mr. and Mrs. Smith. And as you can see, in Mr. and Mrs. Brown, blood type O do not appear. But in Mr. and Mrs. Smith, blood type O appear. So it means that Mr. and Mrs. Smith is the parents of baby number 1 with blood type O. Now, some traits are controlled by sex-related inheritance. In humans, sex is determined by the XX chromosome for female and the XY chromosome for males. Sex-related inheritance can be categorized in 3 ways. First, we have here the sex-linked traits, which are determined by the genes located in the sex chromosome. Are traits genetically determined by an allele located in the sex chromosome? So a sex-linked trait is a trait that is controlled by a gene or an allele located on the sex chromosome. In humans, the sex chromosomes are the X chromosomes and Y chromosome. A trait that is determined by the allele on the X chromosome is particularly described as X-linked, whereas that determined by the allele on the Y chromosome is said to be Y-linked. Males have only one X chromosome, thus, if they inherit the affected X, they will have the disorder. Females have two X chromosomes. Therefore, they can carry the trait without being affected if it acts in a recessive manner. Here are the examples of sex-linked traits. Number one, we have the red-green color blindness, or simply the color blinds. We have also the hemophilia, or considered the bleeders. And we have the Duchenne muscular, and the other one is a Haley ears, which is located in the Y chromosome. Now, let's cross a non-color-blind man and a color-blind woman. What is the chance that the son will become colorblind? So try to look at the cross here. So based on the cross, we have here the female, which is a colorblind, and a male, which is a non-colorblind. So when we cross this, okay, here, the female, the two females are considered colorblind gene carriers, and then the male here is considered a colorblind. So with that, we can say that the genotype is 2XZ for the female, and then 2XCY for the male. That represent a colorblind, and for the female, that represent a a daughter, which is a color blind gene carrier. Okay, so do you understand the cross between the sex linked traits? Now, let's proceed to the next one, and that is the sex influence traits. So, what is that sex influence trait? It occurs when the phenotypes are different between males and females with the same genotype. The examples are male pattern baldness, though the condition is not restricted in males, so this gene has two alleles, bald and non-bald. The behaviors of the products of these genes are highly influenced by the hormones in the individual, particularly by the hormone testosterone. All humans have testosterone, but males have much higher levels of this hormone than females do. The result is that in males, the badness allele behaves like a dominant allele, while in females, it behaves like a recessive allele. Another example is length of the index finger, the body hair, and muscle mass. Now, let's proceed to the third one, which is the sex-limited traits. These are traits that can only be expressed in one sex or the other. Sex-limited traits are generally autosomal, which means they are not found in the X or the Y chromosomes. Sex-limited traits are those that are expressed exclusively in one sex. However, sex-influenced traits are expressed in both sexes, but more frequently in one than in the other sex. Examples of these are the genes that control milk yield and quality in dairy cattle. For example, are present in both bulls and cows, but their effects are expressed only in one female cattle. Another example of the sex-limited traits is the beard in males, barred color or coloring in chickens, normally visible only in the roosters, and secondary hormonal development. I hope you learned a lot in non-Mendelian patterns of inheritance. Again, this is teacher Jeanette saying goodbye and see you next time. Bye bye.